Hi, in this video, we're going to explore how to do upstream analysis of bulk synthetic libraries. So here I already have a prepared project with some of the samples here. Actually, I have four samples coming from 3 rounds of enrichment with phage displays. So to start the analysis of amplicon data of synthetic libraries, we're going to use MiXCR Amplicon Alignment block. So let's add this block to our project. And what we see here is the settings, obviously. So let's select our data set, and then the main place where we put our reference, the synthetic reference, in this text area. Here we can actually put just one sequence, for example. Yeah, and then the alignment will be done against this sequence. This sequence should be a nucleotide sequence of the BDG region of our construct. It's important that this sequence is in frame, it doesn't have, some weird letters, etc. So, and it should be exactly BDG region. We can also have multiple reference sequences, and we can provide multiple sequences here using the standard FASTA format. So once we have our reference provided, we need to select the chain. Well, in most cases, if we are taking the antibody, then it's monoglobulins, primarily heavy, but sometimes TCRs or light chains, so it depends. IG heavy is the common chains. And then I click run. So this will trigger the analysis. And here under the reference alignment logs, we actually can see the row logs of the library construction. So here actually the underlying tool of the block will reconstruct the reference, the V part, J part, and to the alignment to extract the CDR3 regions and the full VG region. And yeah, once the analysis is done, we can go to the individual sample and see the individual rigorous reports provided and the log of MiXCR execution with lots of information. But the overall overview of the quality is already here. You see the alignment rate. The green means the proportion of the reads that were successfully aligned, while yellow, red, the reads that were not aligned. And here you can see what are the reasons. Like, for example, for yellow, no gene hits, meaning that this portion of reads was not aligned to our reference. Before we proceed downstream, it's important to note here that there are some advanced settings that we can. The first one is the tag pattern. By default, we assume that the data does not have any barcodes, like unique molecular identifiers. But actually, if our data has unique molecular identifiers, then we can put the tag pattern here using the MiXCR syntax to, for example, let's say I'm just playing around to show how you can put like the UMI at the beginning. So this means the beginning of read one, then the UMI, then everything which follows the 12 nucleotides of UMI will be considered as read one, and then everything. This is the read separator. If you have a paired end data, keep in mind that we can have single end data. Yeah, in this case it's paired end, so this is a read separator, and everything which goes after backslash is actually the read two. So from the start of read two to the end of read two is what we consider as read two. With this tag pattern for click run, the first 12 nucleotides of read one will be used as a unique molecular identifier. And then downstream, the abundance metric used for our antibodies will be UMI count. But I'm not going to love this because this data do not have reference. There are other advanced settings actually here. The one is error correction. Error correction is a very, very important part of MiXCR processing. At the hood, it corrects for sequencing and PCR errors. And for synthetic libraries, for artificial libraries, it's very, very frequent, like the random mutagenesis of the library to introduce more variability, more diversity in the library. And with the typical synthetic libraries, recommend using the relaxed error correction because it will lead to a faster assembly. If you know that your quality of sequencing is not that great, or I don't know, you use a long read technology which is known to be error prone, then it's better to use the default MiXCR error correction, which will take more time to for the analysis, but well, it will definitely clean all of the sequencing and PCR errors. Yeah, and finally, well, in extreme cases, if you want to do really fast, you can just turn off error correction, but keep in mind this will result in some of the erroneous sequences, erroneous clonotypes. As usual, when we do the upstream analysis with MiXCR, we recommend to put some number here, take only this number of reads into the analysis, like 1000, for example. This will run the analysis on the 1000 of reads just to do the quick, dry run for the whole pipeline. Yeah, and if you run on the cloud or HPC sometimes, and your libraries are really big and you want to ensure that there is enough memory to keep to the process, you can override these defaults, but that's a very advanced thing. So you don't do this normally. So once the analysis is complete, we already can proceed to the downstream analysis. The first thing is just to, for example, look on our clonotypes, on our antibodies, annotated antibodies with clonotype browser. Just for example, I'm selecting our data set, and we have here all of our antibodies. If we go to the overlap, then we will have not the first sample, but the overall view of all of the clonotypes that we have. Yeah, and here we have the VDJ region. I mean, is it a sequence with the VDJ region? That's what is expected in this block. The assembling feature is VDJ region. So all of our protein sequences are in frame, no stop codons, no any other things. So very convenient. That's all for today. Stay tuned for the next guides.